In designing a study, epidemiologists have to take into account the time and resources they have available. But they still have to make sure they collect data that's as accurate as possible. That is, that they've minimized all possible biases. It's important for them to choose the study design that's most practical and appropriate to the situation. So now we'll discuss different types of study designs, when each should be used, what kind of information each provides, and the benefits and shortcomings of each. Experimental studies, also known as intervention studies, are one type of study design. They're usually done to test a new treatment for a disease, such as the Salk vaccine for polio. In these studies or clinical trials, an investigator exposes the experimental group to the treatment, the vaccine in this case, then follows study participants over time to assess outcomes. Intervention studies are conducted very much like laboratory experiments and usually involve random assignment to one intervention or treatment group and another comparison or control group. Subjects are assigned at random to equalize the groups with respect to bias, factors that could influence outcomes. The control group receives no intervention or whatever is the current standard treatment for the disease. Actually, to minimize bias, the control group is given a placebo, something that looks like the drug or vaccine being tested, but which is an inactive substance. Clinical trials are usually double-blind, which means that neither the doctor nor the patient knows whether the patient is receiving the treatment or a placebo. This too is done to minimize bias, which could influence outcomes. The clinical trial, also referred to as a randomized control trial or RCT, is the gold standard study design, meaning it's the study design that gets the most accurate data. In many cases, public health studies can't utilize the RCT as a study design. In those cases, epidemiologists and public health researchers most often rely on observational studies. The next three study designs we'll discuss, that is prospective cohort studies, retrospective cohort studies, and case control studies, are all observational studies. First, we'll look at the cohort studies. Think about a study that could identify risk factors associated with cardiovascular disease, or CVD. The design that would most accurately identify CVD risk factors is a prospective cohort study. In this type of study, investigators test a hypothesis. They may identify individuals with specific exposures, such as smoking and lack of physical activity, and study these individuals over a period of time to see if they develop CVD. In fact, we're talking about an actual study called the Framingham Heart Study, which began in 1948 and continues today. At that time, little was known about the general causes of heart disease and stroke, but CVD death rates had been increasing and no one knew why. The purpose of the study was to identify common risk factors for CVD. A large group, 5,209 men and women ages 30 to 62, from Framingham, Massachusetts, was recruited. Everyone received physical examinations and lifestyle interviews at the beginning of the study. Eligibility required that they show no signs of CVD. They were followed with exams and lifestyle interviews every 2 years over a number of years to see who developed CVD. A second generation was enrolled in 1971. The Framingham Heart Study led to the identification of the major risk factors for CVD: high cholesterol, high blood pressure, smoking, obesity, and physical inactivity. Unfortunately, prospective cohort studies are costly and time-consuming. The retrospective cohort study is an alternative design. In a retrospective cohort study, both the exposure and the health outcome under study have already occurred. Basically, participants are selected on the basis of having had the exposure. They are followed for the incidence of disease up to the present. In order to identify the presence of exposures as well as the incidence of disease, existing records are used, including employment records, medical records, vital statistics records, and disease registries. While prospective cohort studies may also use records, prospective cohort studies are further informed by data directly collected from participants through interviews and physical examinations. There are both advantages and disadvantages in selecting a retrospective design versus a prospective design. Because the outcome has already occurred, investigators in a retrospective study don't have to wait until the disease or outcome develops over time. This is extremely important for studying diseases such as lung cancer that take a long time to develop. Also, the use of existing records costs less money than collecting primary data directly from study participants. The disadvantages are that the ability to explore risk factors and outcomes is limited to existing data and data that is less complete. The last observational design that we'll cover is called the case control study. Unlike the cohort study in which participants are selected based on their exposure status, participants in case control studies are selected based on disease status. Investigators go back in time to assess patients' exposure status. Cases, or those with the disease under study, are often identified using disease registries for birth defects or cancer, for example, medical records, or vital statistics records such as birth or death certificates. Controls, or those without the disease under study, are selected from the same source population so that they're similar to the cases, except for having the disease. If cases are identified from the hospital or clinical records, controls may be patients from the same facility but without the disease under study. Case control studies are efficient for studying rare diseases since the sample size does not have to be as large as that for a cohort study. Remember, participants are selected based on already having the disease. It also allows investigators to study the association of the disease with multiple exposures. Case control studies are efficient when the disease under study has a long latent period, meaning that it takes a long time to develop after possible exposures under study. And similar to retrospective cohort studies, case control studies are less expensive and less time-consuming than prospective cohort studies. The disadvantages of conducting a case control study is that it is inefficient for rare exposures. It's also susceptible to the biases we discussed earlier, namely interviewer and recall bias. Furthermore, it's difficult to assess the time sequence between exposure and disease. This is because cases are identified based on participants having the disease, and investigators have to go back in time to assess exposure. Therefore, they don't always know whether the exposure occurred before or after the disease. All of the study designs discussed so far are analytic studies. That is, randomized control trials which are experimental, and cohort studies and case control studies which are observational are all examples of analytic studies. If you remember from earlier in this lesson, epidemiologists conduct both analytic studies and descriptive studies. To refresh your memory, the purpose of analytic studies is to advance understanding of what causes a disease and to understand the question why. Now we're going to discuss descriptive studies, which describe the health status of populations according to person, place, and time, and to answer the who, what, when, and where of a disease. This lecture will only cover cross-sectional descriptive studies. A cross-sectional descriptive study looks at the association between exposures and outcomes, that is, symptoms and diseases, at a single point in time. It uses surveys, which may be collected using mailed questionnaires, telephone interviews, online surveys, or in-person interviews. There are no follow-ups conducted. The biggest advantage of the descriptive design is that it helps to identify urgent public health and medical resource needs. Descriptive studies are often quicker and less expensive than analytic studies. Ideal as preliminary studies, they generate hypotheses that are often followed up by analytic studies. A disadvantage is that they can't calculate incidence, only prevalence. That is, the total number of existing cases. Also, it's not always clear whether the exposure caused the disease or the disease caused the exposure. For this reason, descriptive studies cannot prove causality, only the existence of a relationship or association between the exposure and outcome. All of these study designs are examples of research. When conducting research, it's a fundamental responsibility of researchers to protect human subjects. In the past, there have been research studies that did not. This resulted in the creation of rules and regulations protecting the rights of human subjects. Examples of human rights violations were the shocking experiments conducted by the Nazis during World War II. In these studies, the effects of high-altitude, hypothermia, experimental medications, and other harmful exposures were studied in concentration camp prisoners. Many subjects died or were permanently disabled as a result. To correct this horrible lapse of protection, the Nuremberg Code was developed in 1949. This code required that voluntary consent of subjects be obtained. The research benefits society. Procedures avoid unnecessary suffering and minimize risk. The risk not exceed the benefit. Subjects be able to withdraw at any time, and investigators stop the study if risks of injury, disability, or death occur. Another key study which failed to protect the rights of human subjects was the Tuskegee syphilis study. The study focused on the natural history of untreated syphilis in a sample of disadvantaged African American men. When treatment became available, it was not provided to the study participants because the investigators didn't want to interrupt the study. Other protections were added as a result of this and other unethical studies, including the Declaration of Helsinki and the U.S. National Research Act. The Declaration of Helsinki stated that consent should be in writing. Research should build upon previous work. Research must follow written protocol. Protocol must be reviewed by an independent committee. Caution must be exercised if the subject is in a dependent relationship with the investigator, and subjects must receive the best proven treatment. The U.S. National Research Act led to the establishment of institutional review boards to review research protocol and ensure that it is ethical and that it protects human subjects. This lecture has been jam-packed with information related to epidemiology. Let's remember that the overarching purpose of epidemiology is to describe disease and related exposures and to identify courses of action. All of the more detailed information in this lesson is what epidemiologists have to consider in ensuring the most accurate data with the time and resources available to them. The TV news, news on the internet, as well as in your print newspapers and magazines, show epidemiology at work. After this lecture, you should be able to identify quite a few.